Chapter 3, Part 3. So when we're looking at this, we are going to start with, we've already looked at Part 1 and Part 2, the cytoplasm and all the organelles, as well as the plasma membrane and everything that makes up that. So now we're going to be looking at the nucleus. So the nucleus is going to be the largest organelle, and what's specific about it, it is going to carry all of the genetic material for our cells. So it is the cell control center. So this nucleus is very, very important. Some nuclei or some cells have one nucleus, and if this occurs, these are going to be called uninucleated, which is this guy right here, one nucleus. However, some have lots of nucleus, multiple. So those are going to be called multinucleated. So they don't have one, they have one plasma membrane, but they're going to have multiple nucleus inside. And then you're also going to have some without a nucleus. And if there is no nucleus in the cell, these are all going to be called anucleated. So an example of an anucleated is going to be a red blood cell. They have no nucleus, but then some we're going to talk about are going to be multicellular. And we're actually going to talk about all three of these types in this semester. So we're talking about the nucleus inside the cell. So the structure of the nucleus, there are three regions inside the cell. The first part is going to be the nuclear envelope. The second part is going to be the nucleoli. And then the third part is going to be the chromatin. So essentially we're going from the outside to the inside. So we're going to start with the first part, which is the nuclear envelope. So if you look down here, the nuclear envelope is the actual outside environment. The chromatin is going to be all the stuff around, and then the nucleolus is going to be this guy. So if you look at it underneath a microscope, you are actually going to see nuclear envelope, nucleolus. So it kind of looks like a dot, a marble inside of a cup. So that's kind of what that looks like. So when we're looking at this nuclear envelope, we're going to take this nuclear envelope, and now we're going to talk about this first region. So when we're looking at the nuclear envelope, some things that we want to keep in mind. It is also a double membrane. So it follows that same pattern as the plasma membrane does. With that double membrane, it is going to have a liquid inside that double membrane. The outside layer of the nuclear envelope is going to be continuous with the rough ER. So we're talking about this outside layer being continuous with the rough ER. So it kind of looks like that, where the inside layer is going to be actually one solid substance. So the outer layer is continuous with the rough ER. The inner layer is going to be called the nuclear lamina. So that would be this guy right here, the inner layer. Lamina means laminated, which means lots of layers. Inside our nucleus, you're going to have these things called nuclear pores. So inside these nuclear pores, they're going to be in the nuclear envelope, and they're teeny tiny little holes. And these teeny tiny little holes are all the way around, and they allow things in and they allow things out. So these nuclear pores allow the substances in and out of the cell. So they kind of act like a transport protein and a plasma membrane, but they're just holes inside the nuclear envelope. So you can see this image here. Here's the nuclear envelope, and all of the little dots are all of the little holes. Inside your nucleus, you're going to have the nuclear envelope. Then you're also going to have the second part, which is the nucleoli. The nucleoli is going to be a small, dark stained body inside the nucleus, and this is where you will have ribosome synthesis. So in this nucleoli section, the primary thing we are going to talk about is going to be RNA and protein synthesis. So to start with that, those are all going to be created by the thing called ribosomes. Ribosomes are going to be synthesized in the nucleoli. So as we could talk, keep ribosomes, keep nuclei, keep nucleolus all in the back of your head. There's usually only about 1 or 2 per cell because you don't need that many because they can knock out ribosomes pretty quickly, but they do exist. So the third part is going to be the chromatin. And our chromatin is going to be our DNA. So it's about 30% made up of our DNA, actual DNA strands. 60% of it are going to be globular histone proteins. And these proteins are going to be essentially the thing that winds the DNA up. And then there's going to be 10% that are going to be in the form of RNA. So these percentages are kind of what makes up chromatin. You guys have all heard of the DNA, but the way the DNA is wound up is going to be specific to these histone proteins that really allow it to do that. So chromatin is going to be formed in these units called nucleosomes. Nucleosomes are the DNA which is wrapped around the histones. So essentially what you do is you take DNA and you add the histone. And so if this is my histone, it kind of looks like a hockey puck. Here's my histone, here's my hockey puck. My DNA is going to wind around it. And then when you have that, you are going to have a nucleosome. So that's kind of how you are going to have this whole thing together. So what we're going to do is we're going to zoom in on this guy right here so you guys can see it. In this particular section, you can see how the DNA is twisted all the way around. This is the form of the chromatin when you are in this structure. When you act, this is just the strand of DNA. So remember, when we're talking about chromatin right now, it is going to be in a strand of DNA kind of like this. But what happens is when you actually go through a process where you have chromosomes, you're going to have something called condensed chromatin. So chromosomes are what we have usually talk about when we talk about DNA, and they are condensed chromatin. So what I want you guys to notice here is that your chromatin is going to be twisted. It's still a helix, but your chromosomes are going to take your chromatin, which looks like this, and they're going to do this. So it is essentially twisted around itself, but the actual strand is still in this kind of twisted form because of those histone proteins in the nucleosomes. But the chromosome is taking that twisted strand and twisting it on itself. So to blow that up again, to look at that image right here, you can see how this guy right here is twisted, but this is where we're twisting. So this whole thing is going to be in its chromosome form, but when you actually look at what is being twisted into the chromosome, this is still twisted first, and then it's twisted on itself. So it's kind of like a double twist. So just want you guys to keep that in mind. Okay, so that's kind of the 3 main parts of the nucleus. What does the nucleus do? Well, the first thing it does is it's going to be responsible for the cell cycle. So the cell cycle is going to be a series of changes, and these series of changes are going to occur for a cell from the time the cell is created or formed to the time the cell reproduces. So it does not say birth and death because cells aren't born and cells don't die. Cells are created and then cells reproduce or divide. So the cell cycle is going to be a series of changes that happen from creation to reproduction. So that is the definition for that guy. There are 2 major phases. One of these phases has a lot of parts, so we're going to talk through that. 2 major phases to the cell cycle. You're going to have one called interphase. And interphase is where you are doing normal cell life. Everything is just kind of happening. You're growing, you're going through normal development, to just kind of doing its thing. And then you're going to go through phase 2, which is going to be cell division. And then this is where the nucleus will come into play. So this is where you'll actually divide into 2 cells. Cell division will have 2 parts. One will be mitosis. One will be cytokinesis. We're going to talk about that in a second, but that's where mitosis actually comes into play. So interphase is the normal cell everyday life. This is interphase. When you look at this interphase, it is actually going to have your DNA in the chromatin state. So it's not dividing. Interphase has subphases, and these subphases are going to consist of a couple things. So here we go. Here's our subphases of our interphase. So the way I like to draw this is I'm going to replicate this image right here. Here is my cell cycle. So this is creation. And this is going to be reproduction. So this is how I'm dividing my cell. Your cell is actually divided into, like we said, we're going to do it this way. Your cell was divided into, this whole thing over here is going to be interphase. So from here all the way around, this is interphase. And then this phase right here, which will be the last part, this will be cell division. So this is kind of how those 2 phases are broken down to. Now we said that our interphase has subphases. So now we're going to break down our interphase into those subphases. So your interphase is going to have 3 subphases. So this first part is going to be called G1, otherwise known as GAP1 subphase. And this is where you are going to have regular cell metabolism. Normal everyday life is happening for this cell. It is not mitotically active. It is synthesizing proteins. This guy can be this length of time, or he could be this whole half over here length of time. He's going to have the most variable time. He can be long or he can be short, but this is just normal everyday cell activities. When you get to G1, G1 will then jump to the next phase, which is called the S phase. And S phase stands for synthetic. So let me do G1 phase down here. S phase is going to be synthetic phase, and this is where you will have DNA replication. So where you will actually divide your DNA. You'll have 2 sets of chromosomes that are attached at the middle. So your DNA will actually go through a division. Talk about this in a couple seconds. And then after S phase, you go through the next part, which is going to be the G2 phase, or GAP2 phase right here. And now that we've divided our DNA in S phase, what we're going to do here is we're going to prepare for cell division. So you're going to get ready to be able to divide the cell in half, because that is the next phase, is this cell division phase. Okay, so a couple cool things about these subphases is each of these subphases, I'm going to skip this DNA replication right here and we're going to come back to it. I don't like the fact that this chapter does it right in the middle of some concepts. So when we get to DNA replication, we're going to do that next. But one thing that is interesting to note is with these interphase phases, there's 2 extra things that are really cool. At the end of G1, you will have something called a G1 checkpoint. And at G1 checkpoint, what the cell is doing is it is making sure that everything in G1 is happening normally and it's good to go. G1 checkpoint has to give the yes before the cell moves into S phase. So it's kind of like a control mechanism to make sure that it is, if there's anything going on in normal everyday life, the cell will stop and it won't create a new cell. You'll also have something called, way back here, a G2 or an M checkpoint. G2 for the end of G2 phase, or M for the beginning of mitosis, which will be the cell division. And this checkpoint right here is actually doing the same thing. If things are not what they need to be, it will stop and there will be no cell division. So it'll actually keep things from occurring. This is a lot of focus on research in the world of cancer, because for cancer, it is a healthy cell that has essentially become unhealthy because it has replicated and created itself too many times. So now you have overgrowth of that cell. During that phase, there's usually going to be mutations that happen, but G1 checkpoint should catch cells that aren't doing what they are doing and limit them from continuing the rest of this phase. G2 checkpoint should also catch cells that aren't healthy and not going through cell division. So there's a lot of cancer focus into this cell cycle and these two checkpoints. Okay, so we've done G1 phase, S phase, G2 phase, the cell division phase. So we're going to drive down past the DNA replication and we're going to come back to it. But in this cell division phase, it's going to be the M phase. M phase is going to be mitosis and cytokinesis. And inside this mitosis phase, you're going to have the division of the actual nucleus itself. And these are the 4 phases of mitosis. So this is where I will defer to lab on the concepts involved in mitosis. So what you need to know is that in cell division, it will go through those same steps. It will go through mitosis. As it goes through mitosis, it will go through prophase. Then it will go through metaphase. Then it will go through anaphase. Then it will go through telophase. And then while these guys are occurring, it is going to go through cytokinesis. And then from the first time the cell is created, as it goes through this cell cycle, at the end of this, you are going to have 2 daughter cells with a nucleus and a nucleoli with all the DNA inside. When it comes to this mitosis and these phases, like I said, I'm going to defer to lab. What you need to know for the particular class for the lecture is you just need to know what these names of the phases are and the order they go in. So prophase will have an early and a late. You do not need to know the image because that is going to be more of a lab. But however, if you'd like to look at these for labs, that'd be great. After prophase, you have metaphase, where this is where you're going to line everything up right down the middle. After you have metaphase, then it's going through anaphase. Anaphase is where it's actually splitting down the center. So here's our center, and we're pulling to either side. After anaphase, you go through telophase, and this is when you are actually splitting the plasma membrane. After that, you're going to have cytokinesis, where you're actually dividing all of the cells. So you can see here how we are recreating that plasma membrane, and you're splitting up all of the other organelles in the cytoplasm in cytokinesis. So when we're doing these whole mitosis, what you need to know for this class is the order of these phases. The details and the images and the pictures and being able to identify them is going to be more of a lab concept. So what you guys just need to know is going to be the order of these mitosis phases that occur right here. Okay, so that is the cell cycle, and that's what is happening. So now what we're going to do is we're going to jump and we're going to do this DNA replication. So I like to do these a little out of order of the slides, so please bear with me, but I think it makes the most sense to do everything topic by topic. So DNA replication. When we're talking about DNA replication, what you are doing here, the goal is to copy the actual DNA. So you are making it the exact same as it is. So we are copying the DNA. This is going to begin at S phase. So between G1 and G2. And it follows a 3-step process. So the 3-step process that DNA replication will follow. Step 1, you're going to uncoil the DNA. So like we said earlier, in this particular, when we're in the chromatin form, we are still in a twisted form around this histone protein. So it's kind of acting like a ribbon on a spool. So it itself is not twisted into the chromatin form, but the actual strand of DNA is twisted around these histone proteins that essentially just keep it all straight, kind of like a ribbon on a spool. So you have to uncoil the DNA to be able to replicate that. So what happens with this is in order to uncoil the DNA, you're going to have enzymes. And you're going to have specific enzymes that will unwind the double helix of DNA. And the reason you're doing this is to expose the bases. Now remember, DNA bases are going to be A to T, so it's adenine to thymine, and C to G. This was covered in chapter 2. Cytosine and guanine. Remember, we are talking DNA. So DNA has A to T as a complementary base and C to G as a complementary base. So we're uncoiling the DNA. So here's our little image down here that we're going to reference. So here's our coiled DNA right here. And so this guy right here is an enzyme. And this enzyme has to unwind and expose the bases. So you can see how essentially he's straightening it out. So he is essentially just taking your coiled, coil, and making it straight. So now we can use this straight line. Okay, so after we have uncoiled the DNA, next, the bonds between the base pairs are going to be broken. And when you break the bonds between the base pairs, you are essentially making it unwind and stay unwound. Sorry, y'all, typing and writing is a little bit harder than you think. Okay, so as we're doing this broken base pairs, how we're doing this is we have our coiled DNA. Coiled DNA, we have just made it straight, which is awesome. However, this right here is still going to be a double helix. So you are still going to have things connected to each other. So what we're saying in part 1 is that we've uncoiled the DNA. That's awesome. Part 2, we now have to break these base pairs and essentially down this ladder rung in order to have a side that is open. So the bonds between the base pairs are going to be broken. And this is going to happen at the replication fork. So when you have this happen at the replication fork, you're going to take this. He's gone from here. This is step 1. Step 2 is going straight. And then step 3 is actually splitting this guy into 2. Where now I'm going to have these guys all open. This replication fork will essentially unzip. So this is your replication fork right here. And he will essentially unzip your DNA. So coming down to this picture right here, you can see we're wound, we go straight. We're going to be at our replication fork. And this is where we're going to break the hydrogen bonds, and you're going to have one strand go up and one strand go down. So that's kind of what has happened here. Okay, so now the next part is after we've broken those hydrogen bonds and we've broken the base pairs, then you're going to go through a process where the enzyme DNA polymerase, so this is the name of the enzyme, DNA polymerase, and what he's going to do is he's going to put free complementary nucleotides attached to the parent end. Okay, that's a lot of information. So he's going to put free complementary nucleotides, but he's going to attach it to the parent end of the DNA. So to start with, we have a polymerase. So this is an enzyme. So we've gone from here, we've gone straight. Now we've split it up like this. When we've split it up, remember this is a double layer, that's why we were able to split it. When we have split it up, we are going to have these open ended nucleotides over here. So if this nucleotide right here was an A, the piece that was attached to it right here is going to be its complementary part. So A to T. This is going to be a T. If this was a C, the complementary part to that C is going to be this G. So you can kind of see how it has unzipped. These guys went together, these guys went together, these went together, these went together, and these went together. So let's just finish that up. We have A, we have C, now we're going to do G. G goes to C. We're going to do T. T goes to A. And so that way all the way down. So when we've unzipped our DNA, what we're saying here in number 3 is we're going to have something called a DNA polymerase. So he's going to be a protein. Sorry, well, he isn't protein, but he's an enzyme. He's called DNA polymerase. And he is going to add free nucleotides to the end. So I'm going to use a different color to show this. So if I have an A right here, my DNA polymerase is going to come down right here, and he's going to add a T, because this is the complementary part. My C right here, he's going to move on down, and he's going to take the C, and he's going to add a G. And then we're going to hit the G, and he's going to add a C, and then he's going to come down and hit the T and add an A. Then what you're also going to do is you're going to have another DNA polymerase who's going to come this way. And as he's doing, that's what we mean when we mean the parent end of the DNA. So he's going to come down this way. And this A, he's going to add to a free complementary nucleotide. That is a T. This C is going to be added as a G. The G is going to have a C added, and the T is going to have an A added. So as our DNA polymerase is adding these nucleotides, they are essentially creating a copy. So at the end of this, you're going to have one strand. That is the original strand, and it's going to be A, C, G, T. So this is this guy right here. And what we've done is we have added this strand right here. So at the end of it, you're going to have an added strand. And this added strand will look like this, and it will be T, G, C, A. So that's what this guy is right here. However, we also had this other strand right here. So the original one is going to be this part right here. So here was my original. And his nucleotides were T, G, C, A. So you can see his right here. And then what the DNA polymerase did is it added in the complementary nucleotides. So to my T, it added an A. From my G, it added a C. For my C, it added a G. And from my A, it added a T. If you look at this, with the black is the original strand, and the red is the new strand. You end up with two exact copies of your DNA. So this is what we mean when we say the DNA polymerase will put free complementary nucleotides attached to the DNA. This is how it's going to happen. Is it's going to come through and it's going to add those complementary nucleotides. So to look up at this image one more time, you can see how we've added, here's our DNA polymerase, and it is adding these free nucleotides as it matches. And then after it does that, it will actually coil back up. You're going to have one up here, and then you're going to have one down here. So you go from 1 to 2. So that is what's happening with DNA replication. So all of this is happening during this S phase right here. DNA replication does not happen during cell division. DNA replication happens during S phase. Okay, and then after this enzyme DNA polymerase will put free complementary nucleotides attached back onto the DNA. Then what it's going to do is it's going to go through a process where it is going to restore. And after this, what it is essentially doing, it is putting segments or splicing segments of DNA together. So with this restoration, everything that we just talked about. Let's see, here's a long strand of DNA, and then this is where we had my DNA polymerase, and then he went back, and this guy was twisted, and then we had this one, and he was doing the same thing. We had another DNA polymerase here, and then he was twisted. So you have to essentially take this guy and splice him together with this guy. So you're just going to go through, you can have multiple places on the DNA going through this process of replication, and then you essentially have to cut and copy and paste it together, kind of like you do with a video. So that's what's happening in that last phase, or that last part to really wrap it up. Okay, so that is the S phase cell DNA replication. We've already gone through this mitosis and these phases that you need to know. Again, just looking at these guys, you need to know the order and kind of the general idea of what is happening. And then we had already talked about the control of cell division. So there's go and then there's stop signals. So to really just hone in on this part, I'm going to talk about these guys right here. So the checkpoints in the cell cycle, here we go. So we're going to have these guys right here. That's not a very good drawing, so let me redraw that. Here's my cell cycle. I'm going to have G1 phase, S phase, G2 phase, and then over here is going to be my cell division or my mitosis. Remember, we said for the checkpoint, there was going to be a G1 checkpoint, which is a restriction point, and it's the most important. And if it does not pass muster, the cell will not keep dividing. And then the other checkpoint is going to be over here with the G2 or the M checkpoint. So those are stop and go signals inside the cell. Okay, so that was DNA synthesis. Now what we're going to do is this last section is going to kind of go into a little bit more nitty-gritty, and we're going to do protein synthesis. So this is the last major section of this chapter. So with protein synthesis, it is similar to DNA, similar. It is not the same thing. So just keeping that in mind, it is going to be a similar process. So with DNA and proteins, how they actually work. So DNA will carry the master blueprint for proteins. So DNA holds the blueprint to replicate itself. DNA also holds the blueprint to replicate proteins. So the way it does that is the DNA will actually code the order of the proteins. So this is very, very, very important because as we talked about in chapter 2, proteins have a very specific structure. If the structure of the protein changes, it will be denatured. So if we change the structure, we change the protein. What we're talking about right now is the blueprint. It's the architectural rendering of that protein. So if you change one little part of that architectural rendering or the instructions, you're going to change the entire protein structure. If you change the structure of the protein, you're going to change its function. So getting the copy of the actual architecture of the code is very, very, very important. Okay, so in the world of the protein, here's some common terms we're going to talk about. A gene. When we're talking about a gene, it is the DNA that codes for one polypeptide. So one string of proteins. So here's my amino acid, amino acid, amino acid, amino acid. Put them together. I'm going to have all of these as peptide bonds. You are going to get a polypeptide. So a gene codes for one polypeptide chain. Humans can have up to 2,500 genes or 2,500 protein encoding genes. So that's a ton. So just keep in mind, we're talking about a lot of information. So with this gene, it is going to be a sequence of 3 bases. And these are going to make a triplet. So when you have a triplet, you're going to have 3 bases. So it's going to be 1, 2, 3, 1, 2, 3, 1, 2, 3. What we're talking about with this is our DNA basis because we are still in the world of DNA. So I'm going to have an A, T, C, A, G, T, A. These are a sequence of 3 bases, and this is going to make a triplet. This is essentially a word. This one word equals 1 amino acid. So an amino acid is going to be the building block that makes up these genes. But we know which amino acid we're going to use is because the triplet essentially is going to be the word that tells us which one we're doing. So sequence of 3 bases is going to make a triplet. And a triplet codes for a specific amino acid. Okay, what you're also going to hear about are some terms that are going to be exons or introns. So just to give you these definitions, exons are part of codes that are amino acids. So we like exons. Introns are non-coding segments. So right now, we do not think introns do anything, so we're going to need to cut them out eventually. It's kind of like the B-roll of our protein building. We're going to need to take it out because it's not important to the story. So those are some terms you're going to use. Okay, so we're going to use the term RNA and DNA now. So we've already talked about DNA. Here's where we're going to kind of differentiate between RNA and DNA. So as a reminder, when we had DNA was going to be double-stranded, whereas when we talked about RNA, this was covered in chapter 2, it was single-stranded. When we talked about DNA, the sugar for the DNA was deoxyribose for deoxyribose nucleic acid. The sugar for RNA was ribose for ribonucleic acid. And then for the DNA, all the bases were the same. You had C and G, but you had A and you had U. You did not, you had A and you had T. You had thymine. But when you got to RNA, your bases were not the same. You had C to G and you had A to U, uracil. So this one changes. So as a reminder, here's a comparison between DNA and RNA. The way it's going to work is RNA is the go-between molecule that essentially links DNA to proteins. So you're going to go from DNA, you're going to go to RNA, and then you're eventually going to make a protein. So it's the in-between instructions. It's the carbon copy in between. As we go through this process, we'll talk about these a lot more in depth. This is an overview. So there are 3 types of RNA: messenger, ribosomal, and transfer. So this is this in-between phase right here. So the 3 types of RNA. The first one is this messenger RNA. With messenger RNA, it is single-stranded, so it's the same shape as RNA. Messenger RNA is really, really, really important because it will be mostly involved with the DNA template to the mRNA. So with the messenger RNA, what it is going to be is it is actually going to be a long nucleotide strand that looks like half the DNA. And the reason it looks like half the DNA is it will be only complementary base pairs. Only complementary base pairs. So it's only going to have one set of base pairs. It's not going to have the connected part. Okay, the second type of RNA is going to be ribosomal RNA. And ribosomal RNA is going to form the ribosome subunit. And when it's got the ribosome subunit, we are going to take this mRNA up here. So this guy right here, he's going to take the mRNA, and he's going to transfer the mRNA into its actual polypeptide. So this guy right here is going to take this ribosomal subunit, and then it's going to take the DNA, the mRNA right here. This is going to be my ribosome. It's going to take the mRNA strand right in the middle of it. This guy together is my subunit. And when I put these things together, I am going to end up getting a polypeptide. So with the ribosomal RNA, the big thing to take away is just that you're going to surround your mRNA strand with a ribosome. When you put those two together, you get the ribosomal RNA subunit. This is what's going to lead us to that polypeptide. And then the most abstract one, in my opinion, is going to be a transfer RNA. So messenger RNA is also mRNA. Ribosomal is going to be rRNA. And transfer is going to be tRNA. So sorry I didn't left that out earlier. So transfer RNA is the most abstract to me, and it is going to be a molecule that transfers an amino acid to the subunit. So what this is going to say is if I have my, let me zoom up. Here's my subunit. So we're just taking this guy and moving him down. If I take this subunit right here, I'm going to take my, it looks kind of like a weird shape right here, and he's going to have my amino acid, which is my 3, my gene, so my coding for those 3, and essentially my transfer RNA, this guy right here is my tRNA, and he is going to transfer this amino acid onto my subunit. So what he's doing is he's combining your transfer RNA with your ribosomal RNA. It's going to make sense in a second when we actually talk about the process, but those are the terms that you just need to know. Okay, when we're going through this next step, we're going to talk about protein synthesis. The whole purpose of this section is protein synthesis. So with protein synthesis, there are 2 steps to protein synthesis. We are creating new proteins. Okay, it involves 2 major steps. You're going to go through the process of transcription. Transcription will occur in the nucleus. So here's my nucleus. Inside my nucleus for transcription, I will have my DNA converted into mRNA. So in my nucleus, I am going to have my DNA converted into my mRNA. To do this, at the first part, I'm going to do my DNA bases. And then this step will go from DNA bases to my RNA bases. Then the next step is going to be translation. Translation is going to occur in the cytoplasm. So if this was my nucleus, here is the rest of my cell. So transcription happens in the nucleus, and it's where I'm going to take my DNA to my mRNA. This mRNA is going to travel outside the cell or outside the nucleus into my cytoplasm, and then I'm going to go from mRNA to a polypeptide. So to do this, this is going to happen in the cytoplasm, and I am going to go from my bases, my RNA bases. That's where I'm going to start, and I'm going to end up with my amino acids. And my amino acids are giving me my protein. So the process of transcription and translation are what give you DNA through a multi-step process to my protein here. So transcription comes before translation. I like to describe it as if you've got a doctor who's got really bad handwriting and they write you a script, then you have to take it to the pharmacy for them to translate it. You have to do transcription, before you can do translation. Okay, so we're going to do an overview of these two processes. So the first one is going to be transcription. With the process of transcription, remember we are going from DNA to mRNA, and this is going to occur in the nucleus. So the mRNA is the complementary form of the DNA. So we're going to transfer the DNA to its complementary sequence of mRNA. So that is what that process is talking about. We're going to do this with something called an RNA polymerase. Do not get that confused with the DNA polymerase. DNA was going to be replicating DNA. RNA polymerase is going to be synthesizing protein, so do not get them confused. But it is also going to follow a 3-step process that is going to be almost the same thing as the DNA synthesis we talked about earlier. So transcription has 3 phases. Phase 1, initiation. With this phase of initiation, what you're doing first is the RNA polymerase is going to pull apart the DNA strand. So we're going from here, here's DNA, and we're going to put an RNA polymerase protein on it, and you're essentially going to get my DNA pulled apart. So that is going to be this phase right here, initiation. You're pulling apart that DNA. You can kind of see it right here. Okay, then the next phase is going to be phase 2, and that is going to be elongation. And initiation, we're now going to do the phase of elongation, so phase 2. We're going to make everything longer. So with phase 2, we've now split everything apart, and the RNA polymerase, remember that, will align complementary RNA, that's important, nucleotides to the DNA bases. And then it will link them together. So what it's actually going to do is it's going to do the same thing that we did in the DNA replication. This little RNA polymerase guy right here is going to come through, and you have all of these nucleotide bases that are now open, and this RNA polymerase is going to come through and add in the complementary mRNA sequence. So it's going to be similar, but it's going to be slightly different. So we're going to break this down real quick. Okay. So here on my 1, 2, 3, 4, 5. If I was going to take my DNA, here's my DNA strand. And on this one, I have an A, C, G, T, A. What my RNA polymerase is going to do, I'm going to shred this out. We're going to A, C, G, T, A. A, C, G, T, A. What we're saying is your RNA polymerase is going to come along and he's going to add the complementary RNA nucleotide. So this is different. This is where it is going to flip from an A connecting to a thymine to A connecting to a uracil. So here's my A. What it will come and put on is U, because A is connected to U, because this is going to be my mRNA. This is my DNA sequence right here. But what I'm actually creating is the mRNA, which has the opposite, has a different nucleotide instead of thymine. My C is still going to connect to a G. My G is still going to connect to a C. My T, DNA has a T. The T will still connect to an A. But the DNA has an A, mRNA is going to have a U. So when you actually get to the final mRNA code, it should be without any T's. All of the T's should be flipped and be U's. So this is how you get that RNA nucleotide sequence put onto the bases. And then it's going to go through a quick step 3. And as it's going through a step 3, it's going to terminate. And essentially what termination means for this is step 3 termination is that the RNA polymerase will essentially hit a stop signal. And so it's going to hit a stop signal. And when it hits the stop signal, transcription will end. So a stop signal we're going to talk about when we get to the next step. What is a stop signal? But your actual code of DNA can code for particular messages. Stop is one of them. So when it actually gets to one of them, it will actually stop. So to look at this image up here, we had initiation where we've done all of the separation. We've done elongation where you are adding the RNA nucleotides. And then you go through the process of termination where once you hit that stop signal, you're going to get a completed mRNA strand, and then you're going to get the RNA polymerase popping off. So that's the process of transcription. At the end of it, what you're going to have is this guy right here. So at the end of it, you're going to have a newly copied mRNA sequence. It is not double-stranded. It is not connected. It is, notice here, it is not connected. It's just doing a copy. This newly made mRNA sequence is now going to travel outside of the nucleus into the cytoplasm, and we're going to go through the process of translation. So that is what we're going to do next. So as we go through translation, so with translation, we are going from mRNA bases to amino acids. And these amino acids are going to code for a specific protein. It's very cool. So for this, each triplet, so each 3 base sequence will be something called a codon. So this is where we're going to bring some of that terminology back in from the beginning. A codon is a 3 base sequence of RNA. So if I have this lovely little RNA right here, my 3 base sequence is going to be this, UGC. If I were to have another one, it would be another 3 base sequence. So this is going to be my codon. There are 4 different kinds of RNA because there's 4 bases. So there's 4 bases. You can have an A, C, G, and a U. And if there's 4 kinds of bases and there's a codon, so there's 3 places it can be, it's 4 to the 3rd power. So there are 64 codon possibilities. So with this genetic code right here, a triplet is a 3 base sequence called a codon. There are 64 possible codons because there's 4 bases and 3 places, so 4 to the 3rd power is 64. So 64 messages or 64 amino acids. All codons mean something. So these are going to be a communication signal. 3 are stop signals. 3 of these are stop signals. 61 will code for an amino acid. 1 will code for start. This one that codes for start is also an amino acid. But if it's the first one, it will say go. So there are only 20 amino acids, but there's 64 codons and 61 code for an amino acid. So of these 20 amino acids, 61 codons, they're going to replicate. So some are going to be redundant. Okay, we're going to come back to our we're going to focus on our tRNA. So we're going to hop back to that topic real quick. Like I said, tRNA is going to be the one of the most complex. It's one of the most abstract. So what you need to know about tRNA is you are going from something called an anticodon. So an anticodon is going to be a 3-base sequence that is opposite the 3-base mRNA codon. So the anticodon, so if I had my DNA and I had it as A, C, G, T, I had my mRNA that was A goes to U, C goes to G, G goes to C, T goes to A. And then I'm going to have my tRNA. So this is my codon. This will be my anticodon. My anticodon is going to be the opposite of this. So U will go to A, G will go to C, C will go to G, and A will go to U. This tRNA, this anticodon, will actually connect to an amino acid. This amino acid will identify these 3 codons right here. So when I say that 61 code for an amino acid, we mean this right here is coding for an amino acid. So tRNA has this weird little shape like this, where you're going to have your 1, 2, 3, that's your anticodon, and it's attached to an amino acid. So if you take this guy right here and flip him upside down, you're going to have your amino acid, and he is attached to a 3-base sequence of A, C, G. So that is how you're actually going to get this tRNA. It's a very complex, complicated system. But translation will have a sequence of 3 phases, just like transcription. So transcription had 3 phases. Translation also has phases. And they're also going to be named the exact same thing: initiation, elongation, termination. So it's a very similar process, but they're going to be named different things. So first part, initiation. So when we're talking about initiation for your translation, what you're going to do is you're going to bind your ribosomal unit or your ribosomal subunit with, that is, this ribosomal subunit is going to have with your tRNA sequence. And essentially what this is going to do is this will scan the mRNA until it hits a start codon. So when it hits a start codon on the mRNA, that way it knows to start. So this is what's going to happen. This ribosomal subunit, which looks like this, is going to have your tRNA hanging out in the middle right here, and you're going to have your mRNA go through, And this is going to keep going through until you hit a start. And once you get to that start, then you will actually start the process. Okay, then you're going to go through phase 2, which is going to be elongation. With elongation, this is going to be a 3-step cycle where you're going to essentially be adding amino acids. You're going to add 1 amino acid at a time. As you go through with elongation, you're going to add this amino acid. The way you're going to do it is you're going to have codon recognition. So as it's going through and pulling this mRNA through, it's got the start signal. Now at this point with elongation, it's looking for the codon. There's going to be an A site. So this is where you're going to actually have the incoming amino acetyl tRNA will bind to the complementary codon and the A site of the ribosome. So to just give a visual, I'm going to zoom on down to this guy right here because it is hard to do it without a visual. So in this part, what we're looking at, oh, it's so blurry, I apologize. So we've done initiation. Here is our mRNA unit. Sorry, here is our ribosomal RNA unit. Here is our DNA, our mRNA strand right here coming in. Inside this ribosomal unit, there is an A site, a P site, and an E site. So what we're saying in this phase of elongation, we were at the first part with codon recognition. What we were saying that the incoming tRNA is in the A site. It's going to start binding to the incoming tRNA is binding to the A site. So that's right here. So it goes up here in my little drawing right here. Inside my subunit, I'm going to have an A site, a P site, and an E site. So what we're saying is that this incoming tRNA is going to bring with it into the A site. Okay, I drew it backwards, so we're going to, yeah. I did my exit side on the wrong side, so we're going to split this. It goes like this. A, P, E. It goes this direction. I was drawing it the other direction, but to match your image, we're going to do this. So what we're saying here is with codon recognition, you are going to add your complementary codon. You're going to add the complementary codon. From your tRNA to the A site. So here is my tRNA. He's going to have an amino acid. And here's my 3 codons. And he is essentially going to come, and the place that he is going is he is headed right for this A spot, right there. That's going to be the first step, is codon recognition. The second step is going to be peptide bond formation. With peptide bond formation, we now have this amino acid in the A spot, and you're going to form a peptide bond between the amino acid in the A spot and the P spot. So in this little picture right here, we've done this. Here's my ribosome. I'm moving my mRNA through. Here's my 3 sites, A, P, E. With elongation, you essentially, as you move this guy through, you just keep adding more and more. So eventually you're going to have your amino acids that are sitting here, 1, 2, 3, and you've put on another one, 1, 2, 3, and peptide bond formation is going to be saying that you are connecting these two amino acids right here. So you're forming a peptide bond between the amino acid in the A spot and in the P spot. Okay, so the third part of this elongation part is after that, you're going to do something called translocation. And with translocation, you are going to have that ribosome move down the mRNA. And as it does, it will move tRNA in the A site to the P site. And then the E site, the thing that was in the P site, will move to the E site, and this will get released, empty. So if we are taking our little picture here, and here is my ribosome, A, P, E. Here's my amino acids. Here's my amino acid. Here's my peptide bond between the two. So what we're saying is that this will happen, and then the next part is as this mRNA strand moves, you're going to have essentially the same thing happen, but now this guy is moving here, and this guy is moving here. And so now what we've done is we've moved your A site to your P site. So here's my A site and my amino acid. And he was attached to my AP. He was attached to this guy. So there's still a peptide bond because we've created that guy. So he's still attached. But when we moved him over, he essentially popped out. But what we also did is we added in a new amino acid that's going in here. So essentially what you're going to end up with at the end of it is an amino acid connected to an amino acid connected to an amino acid. All of these are going to be connected with peptide bonds. And so this is going to be continued all the way through. You have a chain of amino acid peptide bonds. So the red is the peptide bond that is holding them together. So this is how you get your chain. Okay, so that was the third step. That was second step. That was elongation. So when we get to the step 3 of our initiation elongation, this is going to be termination. And what is happening with termination is when you hit one of the 3 codons for stop and it enters the A site, it ends. So this will continue, this whole process, until stop codon enters the A site. And then when the stop codon enters the A site, the whole thing is done. So stop codons are going to be UGA, UAA, or UAG. When one of these 3 codons enters the A site of the ribosomal subunit, the whole thing is going to stop. This is essentially the period at the end of the sentence. And what it will do is it will cause the release of the polypeptide chain, and it will separate the ribosome subunits. So if we're going to zoom all the way into this image right here, we had our initiation, our elongation, we had our codon recognition, our peptide bond formation, and our translocation. Remember, all this is doing is saying that your tRNA is going to the A site, then it's going to the P site, and then when it hits the E site, you release your tRNA and you have this polypeptide chain. So with this polypeptide chain, once you've got that, then you're going to hit the third phase when you hit a stop codon, and you're going to essentially break off and release this guy. So that is the process. So focus figure 3.4 is a big important figure to do for translation. So make sure you are reviewing that. We're not going to go through and do rough ER in protein synthesis, so you guys can go ahead and just move on past that. We're not going to go through that. If you want to be able to code DNA, this is going to be an important slide to look at. So for this, just as a reminder, this is your DNA. Then go from your DNA to your mRNA. When you go from DNA to mRNA, this process is going to be transcription. And then your mRNA is going to go to connect to a tRNA. This is your codon. He's going to connect to an anti-codon. That tRNA is going to connect to a polypeptide. This process is translation. So if you want to see how the whole thing works from a DNA sequence, this is something that you need to review, figure 3.34, I think. Okay, and then the last thing we're going to talk about is, for a term you need to know, is going to be 3.12, apoptosis. When you see the word apoptosis, it's a very important word. It's also known as program cell death. And apoptosis essentially means that you are going to destroy the cell can self-destruct. So it can destroy itself. So apoptosis knows when things need to occur. This is a good process for the cell to be able to self-destruct. Sometimes you want it for cancer cells, infected cells, simply old cells, for them to just simply self-destruct. So they're going to do this by leaking chemicals that essentially going to cause them degradation of the DNA. That DNA is going to lead to cell death. So apoptosis, as programmed cell death, essentially means that the cell has the ability to say, my function is no longer working, I need to destroy my structure, and it can actually turn itself off by killing itself and self-destructing. So that's really the focus of 3.2, is just knowing that term of apoptosis. So looking at chapter 3, part 3, it's pretty heavy. Know the cell cycle, the general structure of the nucleus and its part, but the primary focus is knowing the process of DNA synthesis or DNA replication, but then also taking that and doing the cell division, knowing the phases of mitosis, and then going into, after the DNA pieces, focusing on how the proteins are synthesized. That process involves transcription and translation. They each have 3 parts: initiation, elongation, and termination. But those are the focus for this particular part of the chapter.